How do you see the world? We are all influenced by what we can and cannot see, by the patterns that catch our eye. The world's great artists and scientists share the same drive to create or uncover what has not been seen before, to transform what could be into what is. What seems impossible to one generation is a challenge to the next, demanding imagination, ingenuity, and above all, persistence. Our challenge is to cure HIV. amfAR's ICSTEM consortium of European researchers is driven by that challenge. And we hope to lead the way to a cure for the deadliest virus of our lifetime. amfAR funded and launched ICSTEM in response to the Berlin patient, the first person to be cured of HIV in 2008. The announcement of his cure after a complex stem cell transplant to treat cancer was historic. In a major milestone, ICSTEM researchers reported just a few months ago that a stem cell transplant patient in London shows no signs of HIV. Another ICSTEM patient in Dusseldorf also seems to be HIV-free following a similar procedure. With these two new cases, we now have good reason to believe that the Berlin patient was no one-off.
Each new instance of a cure represents an important step forward. With each new case, we can look for patterns that provide critical insights into how a more scalable cure could work. For example, we have scientists at the amfAR Institute for HIV Cure Research who are conducting a sophisticated immunotherapy trial. We have others who are taking a gene therapy approach that kills the virus, kills the reservoir cells, and it uses molecular scissors to remove the genetic instructions for the virus.
We're never going to end the global AIDS epidemic without a cure. And the only way to get to a cure is by making smart, sustained investments in cutting-edge science. At amfAR, we're 100% committed to curing HIV, and we won't rest until we've achieved that goal. In just the last 4 years, through our Countdown to a Cure for AIDS, we've supported research conducted by more than 270 scientists working at close to 100 institutions in 16 countries. Today, a cure may be within our reach. Now is the time for us to join together, to transform what could be into what is, and put an end to this deadly epidemic.
Hello, everybody. Thank you so much for joining us for our inaugural live-streamed Meet the Scientist event. My name is Rowena Johnston, and I'm Vice President and Director of Research here at amfAR, the Foundation for AIDS Research. And first of all, I want to thank you all so much for taking the time to join us for this very special Meet the Scientist virtual program. We at amfAR really appreciate everything that you do to support the work that we do. And our program is special because it's organized in honor of World AIDS Day. And World AIDS Day was created in 1988. It's an international day dedicated to raising awareness around the AIDS epidemic and to remember those who have died of the disease. And it reminds us why our work is so important. There are 38 million people currently living with HIV, and they're counting on us to end this pandemic through research. I'll be talking to 2 amfAR grantees who'll be describing the work they're doing, and you'll have an opportunity to submit questions via the chat feature, and we'll get to those questions during the Q&A session. And today our discussion is ultimately about one of the greatest scientific challenges that we face, and that is curing HIV. And so the question is, why is it so hard to cure HIV? And the answer in a nutshell is the reservoir. Now, I have a picture here of the reservoir. And on the left of your screen, you see a picture of a cell described as active infection. And inside that orange outline of the infected cell, you see a whole lot of processes percolating. And what ends up happening is that that cell produces a new virus that pops out. You see it labeled there as progeny HIV. And the key point here is that that process, right where the number 4 is, when the virus is coming out of the cell, that infected cell becomes visible to the immune system, and the immune system at least has a fighting chance to destroy that cell. On the right-hand side, you see a cell labeled latent infection, and that cell is infected by HIV as well. But the HIV is not undergoing that active replication process. And you see that it has nothing popping out of its surface. And the immune system has no way of knowing that that is an infected cell. It can't move in to destroy that virus. And we have, as I mentioned, two of our great scientists today who are going to talk about their amfAR-funded research on NK cells, or natural killer cells. Now, during this COVID time, we've all become experts on the immune system. And I have a picture that we should have a look at here. So we've been hearing a lot about the immune system in the news. And on the left here, you see a killer T cell shown in red. And the killer T cell's job is to kill infected cells. And it does that by producing kind of toxic bubbles of perforin and granzyme shown in this diagram here. Now, one of the things we have heard about in COVID is if the immune system gets too active, if too much of this is going on, we have this inflammatory process that can be dangerous if the immune system's not regulating itself properly. And then in the middle, in green, we have the B cells, and they are priming. You know, they're basically learning how to respond to the virus. And they turn into antibody-secreting B cells. And now, for sure, the most recent news about vaccines, and we've all been really closely following what's happening with antibodies in COVID, also very important in HIV. And on the right-hand side, we see natural killer, or NK cells. And to be honest, when was the last time you ever heard anybody talk about NK cells? They don't get very much attention. And if you're old enough to understand a Rodney Dangerfield reference, you'll know exactly what I mean when I say they just don't get no respect. And that's a shame, because they actually do a bunch of really cool things that other immune cells don't do. We have, finally, a picture of one of those very cool things that NK cells do. What we're looking at here in yellow is two NK cells, and they're very efficient killers of cancer cells, which we can see here in red. Now, those guys kind of look scary. They look like something out of a sci-fi movie. To be honest with you, if that red cell were an HIV-infected cell, these are kind of the good guys. These are guys we want on our side. And our two grantees are going to be telling us today about the efforts that they're making to see if we can get the NK cells to come in and join in the fight against HIV. So I'm going to introduce our researchers now. As you see, we're joined by two grantees. Dr. Tim Schacker is Vice Dean for Research at the Medical School and Director of the Program in HIV Medicine at the University of Minnesota in Minneapolis. And we're also joined by Dr. Jerome Zack, Distinguished Professor and Chair of the Department of Microbiology, Immunology, and Molecular Genetics, and co-director of the AIDS Institute at UCLA in Los Angeles. Guys, thank you so much for joining us tonight.
My pleasure.
Great to be here, Rowena.
So, Tim, I think we should start with you. You just finished a clinical trial that was funded by amfAR.
Right.
Could we start with just an overview of what was the concept behind that clinical trial?
Sure. So you just laid this out very nicely. The single biggest problem that we've got with curing HIV is this concept of latency. So the virus can be dormant and then occasionally reactivate and make new virus. It's very similar to what we think about with herpes simplex, for example. Once you're infected for life, and there the issue is latency as well. So what's the difference between herpes and HIV? Well, one issue is that with herpes in the norm, or in the person who's not HIV infected, their immune system works pretty well, and it can stop that virus from, when the cell reactivates, it can stop it from making new herpes viruses. In HIV, your immune system, even in people who are on antiretrovirals, your immune system still doesn't work that well. And occasionally that cell can reactivate from latency, and you don't have the machinery in place to take those cells out. Well, NK cells, natural killer cells, are the body's first line of defense against virally infected cells, against tumor cells, and that sort of thing. And so when a virus causes, or when the cell reactivates and the virus is being made, it's possible that NK cells could come in and clear that cell, kill that cell, natural killer, just like they do for tumor cells. The problem in HIV and that's just really unique is that because of HIV infection, NK cells don't work as well. So their systems for clearing viral or tumor cells don't work. So what we thought, we would build on the experience that we have here in Minnesota at the university for using NK cells in cancer therapy to see if we could do that with HIV infected people, because the concept is really kind of the same. You've got a tumor cell, you got to clear it. You've got a cell that's got HIV in it, you got to clear it. And so that was the rationale behind the study. And we wanted to ask what would happen if you gave people with HIV infection, well suppressed on antiretroviral therapy, if we gave them healthy NK cells. So our study design was to go and find a relative of the person who was infected, take their NK cells, amplify them, make more of them, and put them into the person and see what the impact was on the viral reservoir.
I want to get to that issue actually about how you chose the people in your study. I want to get to that in a minute, but I want to go back because I think it's kind of fun and important aspect of NK cells. When you said they're natural killer cells, right? But we know that we also have killer T cells. So what is the difference? If killer T cells could kill HIV infected cells, why would we need NK cells? Can we parse out a little bit the difference between what they do?
Yeah, the difference is the speed at which they work. NK cells are designed to be the first on the scene and to just sort of take out the bad guys. And, it's sort of a broad or a large hammer, if you will. The CD8 T cells, the killer T cells, those are specific for a pathogen. They're specific for HIV. And it takes time for that response to evolve. So, the first kids on the block, if you will, are the NK cells, and they work rapidly, and they have a very expansive way of doing things. Natural CD8 T cells, killer T cells, CD8 T cells, they're very specific for HIV infected cells, and you have to have time for that response to evolve.
And just to clarify, just to get it out of the way, this was a question I had before when I was talking to somebody about this program. The killer T cells are not the same T cells as the ones that get infected by HIV. That's a different kind of.
That's absolutely correct. What gets infected by HIV are CD4 T cells. The way I explain it to my patients is that CD4 T cells are the orchestra conductor of the immune system, and HIV preferentially infects those cells and kills those cells, a variety of mechanisms. And as those cells go away, it's just like an orchestra conductor becoming less efficient and walking off the stage. And you can just imagine how that symphony becomes very disharmonic. So they're different than CD8 T cells.
Right. All right, so let's get back to that issue about, and now you mentioned that for the participants in your trial, you wanted them to be relatives. So you have people living with HIV, and then you needed to find people who were relatives. So like how complicated was that to do? Because basically you're enrolling not just one person, but two people in each case. So, that complicates things, I imagine.
It turned out to be relatively complicated, but the reason we did that is we wanted the cells that we were putting into the HIV infected person to be as close as possible to the HIV infected person. So we wanted their immune systems to at least have some identical features. But the complications of it were, just finding people who were willing to go through the procedures to harvest the NK cells. And, HIV is still a disease that has a lot of stigma to it. And a lot of the people that we screened, frankly, they weren't out to their families. Their relatives didn't know. And then sometimes there were screen failures and that sort of thing. So it turned out to be a relatively complicated process. So it took a while to get everybody enrolled.
So finding this person who's closely related, this is the same kind of thing, we hear about if you're getting an organ transplant. You want somebody who's kind of closely related. The same is true for NK cells?
Similar.
Yeah. Okay.
Yeah, it had to be a mother, it had to be a brother, a sister, mother, father, someone closely related.
All right, but in the end, you did find 8 pairs, I believe.
Yep.
All right, well, we're going to get to that in a minute. So you could have tried any number of things in these patients. What made you think that NK cells was something that you wanted to work on?
Well, again, it's the experience that we've had here in Minnesota with using NK cells in blood-borne cancers, so leukemia, lymphoma, that sort of thing. And what we've shown here over the years is that if you take somebody with leukemia, a blood malignancy that just doesn't respond to therapy, and if you give them these NK cells that their body recognizes, you can take a significant number of people who don't respond to therapy, and then they will respond to therapy. And so we thought, is there a similarity between HIV and cancer treatment? So what would happen if we gave these folks with HIV infection the NK cells in a way that's very similar to how we do it with cancer? So that was our thinking behind this, but also knowing that people with HIV infection have NK cells that simply don't work.
Yeah.
And so those two facts went together.
It's crazy how many similarities in the end we are seeing in how cancer and HIV manifest, and also maybe how we can treat them. I think when we get around to talking to Jerry, we're going to hear more about that too.
Yeah, absolutely.
So NK cells, one of the things, it's true that they swoop in really fast. They're kind of the first responders to the viral emergency, if you like. But they also don't stick around for very long. They're really very short-lived cells. And so that was a challenge you had to overcome as well. How did you decide to go about that? So
in the cancer world, again, what we've done is use a cytokine called IL-2. And as many folks know, we've used IL-2 experimentally in HIV infection to see if we could prolong CD4 T cell life or increase CD4 T cells, et cetera. But one of the things that IL-2 does is that it really stimulates NK cells to, duplicate, live longer, that sort of thing. So when we started this trial, we were using this cytokine called IL-2, which is commonly used in the treatment of some cancers and malignancies. After we did our first couple of patients, there's another molecule that became available made by NantQuest or Immunity Bio in California called N803, which is another cytokine called IL-15. The advantage of IL-15 over IL-2, the advantage of this cytokine over that cytokine, is that it can stimulate CD4 T cells, and so if they have latent HIV, maybe they'll reactivate and make HIV. It stimulates CD8 T cells to expand and become better killers, but it also stimulates, and this is probably its greatest effect, it stimulates NK cells to be more active. And so we switched from IL-2 to IL-15 that prolonged the life of the NK cells, and we think that made them more active against HIV.
All right, there was a lot that you just said in there, so I want to unpack some of it. I mean, first and foremost, what I'm hearing is that you wanted the NK cells to live longer because you need them to do more of their killing that they do. But it's an intriguing idea that doesn't make sense when you first hear about it, that you actually want these latent cells to start making HIV. Because on the one hand, you could sit there thinking, well, if the cell is latent and it's not making HIV, then maybe that's a good thing. Why not encourage that? And that is an approach in HIV cure research. But this approach that you're talking about is one that people call shock and kill,
right,
where you actually want the HIV to come out. So just tell us a little bit more about that. Why is it a good thing? You know what, I think I might put that picture back on the screen. Because I feel like maybe that might be helpful. If we take a look at this picture here and talk through what the advantage is of making the cell active.
Yeah, so if you look at the number 4 on that picture on the left, you'll see the little dots on the surface of the cell. I think that what that's showing are the proteins that the virus makes. It's parts of the protein that appear on the cell surface. Well, that's the target for NK cells. That's the target for CD8 T cells. If those proteins or if those targets aren't there, those cells can't clear that infection. they're that cell. If you look over on the right under latent infection, you'll see that there's none of those dots that represent pieces of the virus or the virus that's being made. So NK cells, CD8 T cells, they can't recognize that. And what happens is occasionally, maybe because you get a cold or the flu or herpes reactivates or something else happens, that cell becomes activated. And when it becomes activated, it looks more like the cell on the left, and you get targets on the cell surface. So the shock and kill method is to say to the latently infected cell, I'm going to make you active, and I'm going to make you look more like the cell on the left, so that the NK cells and the CD8 T cells can find you and kill you.
Right, and the reason we want to do that is because if you stop taking antiretroviral therapy, if you're not adherent, then that latently infected cell can turn into an actively infected cell. It can spread infection, and all of a sudden you're kind of back to where you started in very short order, actually.
Right.
So I'm leaving this picture on the screen now. I don't want to make this all about COVID, but I do know that people have been seeing a lot of COVID news. And actually that progeny HIV it has spike proteins. We've heard a lot about spike proteins in COVID too. You know, it is a common feature in viruses that have an envelope, which HIV does and coronavirus does. And it's exactly those spikes that make it visible to the immune system. I just thought, in case that's helpful to anybody listening, just to draw that connection there between, you know, HIV has spikes, coronavirus has spikes. Those are kind of the magic ingredient that we want the immune system to see so that it can be active.
There has to be a target.
There has to be a target, right. Okay, so you used IL-2 and then you used IL-15. And IL-15 you thought seemed promising because it would not only make the NK cells live longer so they could do more killing, but also because you thought it might make the HIV cells turn from latent into active, and therefore there'd be something for the immune system to see.
Right.
All of this leads us to the big question. How well did your trial work?
So this was a trial that was first designed for safety, because this is the first time we've ever used this approach in an HIV-infected person. And, you know, the HIV-infected people, thank heavens, in 2020 are much different than years ago. And so we have antiretroviral therapy. People are, you know, living longer, approaching normal lifespans, et cetera. So the risk that we can take is much different than we could have 30 years ago, for example. So the first goal was to show that this was safe and that we weren't doing harm. But then, you know, secondarily, we wanted to know if we had any impact on the reservoir, if you will. So in all of our patients that we put onto the trial, this turned out to we demonstrated this was a very safe approach, that there were no significant adverse events from either the infusion of the NK cells from the donor or from the cytokine therapy. But also in all of the patients that we looked at, if you just confine yourselves to peripheral blood, and that's an important distinction, but if you just look in peripheral blood and measure the frequency of cell with a virus that can, you know, a cell that can reactivate and virus that can be made, we saw a significant reduction in the frequency of those cells using the assays, you know, that were available to us. We're confirming that now with other assays, but it's a very promising preliminary result.
Oh, that's kind of fun. Now, I know that I lifted some images that you took from some of your participants. I want to show people some of what it kind of looks like when you do some of the work. So let's take a look at this image right here. This is actually a combination of an image that you made and then one that I made just to kind of explain things. I'll just start out by saying that the guy on the left has an arrow coming out from one of his inguinal lymph nodes, so one of the lymph nodes in his groin. Right. And I know that you took samples from there, and then maybe you can take it away from here. What are we looking at?
Sure. So, you know, the area that we are most interested, or the part of the body that we are most interested in, is the lymphatic system, or which is where most CD4 T cells in the body reside, 94%. And so that's where you really have to be studying to understand the impact on the reservoir. And what I just described was a result in peripheral blood. This is our first attempt to look in the lymphatic tissues to see what the impact was. And what you're looking at here is a lymph node from one of our patients. And all we're doing here is identifying NK cells. So we used a special stain to identify NK cells. And all I'm doing here is showing that there's an increase in the number of NK cells in the lymph node after the infusion. And so what we can tell from this is that the NK cells appear to be getting to where we want them to be, where the main reservoir is.
So you're infusing them into the blood,
Yeah.
if I'm understanding correctly, and you're hoping that they'll migrate to the place where, as you said, most of the CD4 cells are, but where the CD4 cells are is also where the HIV is.
Correct.
And just, it's kind of, I used to do this work too, but it's kind of crazy and cool that you took lymph nodes out of people and you cut them incredibly thin. And I put a note on here that each of these slices is only 4 microns, and you would need about 15 of them stacked together to make even the width of a human hair.
Right.
And all of the brown spots here are NK cells, and they might be NK cells that you infused into the person as part of their treatment, but it might also be some of them might be their own NK cells.
Correct.
So this is a little bit of a mix. All right, just to get that straight. So I'll stop sharing that image. There was another image that you created that I was particularly struck by, and I want to show people that too, because here you're comparing we're looking at a couple of different things, but you're looking not only at a lymph node, but also the gut. And the gut is also an area that has a lot of immune system cells and HIV.
Yeah, no, actually the largest lymphoid organ in your body is the gut. It's the primary line of defense, actually. So all I'm showing you here is that we were able to come up with a way to identify donor cells, donor NK cells from your own NK cells. And so those cells that are staining brown are the donor NK cells on the left in the lymph node, on the right in the gut. And the remarkable thing is that this is 21 days after we infused the NK cells. And in the world of cancer, they've never looked in this amount of detail. And this is really remarkable that you would see these healthy NK cells persist that long in the tissues. Now, this is we've got a lot more work to do. This could just be, we happen to get the right patient at the right time. Maybe other patients, it wouldn't be this persistent, or maybe they will be. This is the beginning of this exploration, but it's really intriguing that we were able to find that.
I think, to me, I don't know if this is a person who received IL-15, but I think it really speaks to maybe the magic juice worked, so to speak, because I know my textbook tells me an NK cell normally only lasts for 2 or 3 days. And you're talking about 21 days. I mean, that's pretty crazy.
Yeah, and in data that I've not shown you, but we just got this data that all of the patients, you're right, without this kind of support, it's 2 days. And in cancer patients, it's not that much longer, but in all of our HIV-infected patients in blood, we could detect these cells out to 8 days.
That is very cool. Congratulations really on getting that trial done, because I know that's a lot of hard work.
It was a labor of love.
It took a while.
Yeah.
So I want to remind our audience, please remember, we want to hear your questions. Our speakers are eager to answer your questions. I'm going to talk to Jerry next. And after we're done talking through Jerry's project, I want to address everyone's questions. Please keep them coming. We definitely want to hear from you. And so with that, Jerry, we do turn to you. Now, as opposed to Tim, who's just finished his amfAR grant, you're about to launch into an amfAR grant. You just got this new funding. And what you're also looking at NK cells, but you're actually looking at NK cells just as they regularly occur. I kind of feel goofy saying, natural killer cells. And you're comparing them to engineered natural killer cells. And there's really a lot of complexity. You've got, kind of a bunch of different ingredients in your research plan that I think are going to take some explanation. But maybe we could start with you too. Just give us kind of the 30,000-foot view of what it is you're hoping to be able to determine with your grant.
Sure. Well, I really appreciate Tim's intro because it really explains a lot of the rationale behind what we're doing. But our lab really tries to model how you could do new therapeutic approaches in HIV. And we do that not by using patients or monkeys, which are the two animals that can be infected with HIV. But we use humanized mice. And so these are mice where we put a human immune system in and then can infect the human cells with HIV, which wouldn't normally infect mice. And we can use those to study different drugs or different therapies, different approaches. And so we've been testing just normal NK cells, normal natural killer cells, right? As opposed to natural killer cells. And when we use this model, we can infect the animals with our virus. We get lots of virus in the blood. We can put them on antiretrovirals, just like one would do with a patient with HIV, and we can suppress viral load. If we take them off drug, we get rebound of viremia, just like you would see in an infected human. So we have a latent reservoir that rebounds. So we've been able to show that we can put normal NK cells into these mice. These are from a different person than made the mice in the first place. And it delays rebound. Okay, so it slows up the ability of the virus to come out of latency, or at least be detectable in the blood.
All right, I'm going to stop you right there. Sorry, Jerry, because I think it really probably behooves us to talk a little bit about the importance of delaying rebound. What might that mean? First of all, what is rebound, and why are we looking for delay in rebound?
Okay, so rebound is when, as Tim explained, if one goes off of antiretrovirals for whatever reason it is, invariably you will get a resurgence of viral replication in the body, which we call rebound. It's a simple term for it. It usually would happen in infected individuals within a few weeks of going off of ART. In our animals, it comes a little quicker, but nonetheless, it's the same phenomenon. If you go off of drug, virus comes back because nothing's stopping it from replicating anymore. If you can manage to slow down or delay when that rebound happens, it suggests you're minimizing the reservoir, because the logic would be the more cells you have in the reservoir, the faster they're going to come out and rebound. Okay, so if you can use delay of rebound, so the timing of rebound is sort of a relative, it's not a very sensitive model, but it's a pretty good model of whether you're impacting the number of virus-infected cells that are in that reservoir. Okay? Great.
Now, I cut you off. You were talking about the 30,000-foot view of your project.
Yeah, so as was discussed, to see, for a cell to kill its target, it has to see something. It has to have a molecular target on the surface. CD8 cells target viral peptides, viral proteins made by the virus itself on the cell. NK cells can do that also, but NK cells also have a bevy, just a bunch of different receptors on them that can target normal cell molecules that indicate the cell is not feeling well. Maybe a simplistic term, but if the cell is perturbed in any way, if it's a sick cell, an NK cell can take it out, whether it's infected with HIV or not, right? So it's the NK cell's main goal isn't to knock out HIV, it's to get rid of cells that are diseased or malignant, all right? So the rationale we took, or we're planning to take, because we're just beginning the project, is to engineer into NK cells a molecule, which we're going to call a CAR, or a chimeric antigen receptor, that would add greater ability of the NK cell to see HIV in the target. Okay, so it, in theory, increase its ability to look towards an HIV-infected cell instead of towards a random cell not feeling well. Okay. So
yeah, your conversation about a cell not feeling well, I actually generated some images for this, and then at the last minute decided, okay, maybe that's too many images that Rowena has made up. Yeah. But I think it's actually a really cool characteristic of NK cells that probably deserves mention here, and that is, we're talking about that the cell has to be actively producing HIV for those particular signals to be visible to the immune system. And another way an infected cell, if a cell is infected with a virus, it can kind of chew up the virus inside itself and essentially run up little bits of it up a flagpole posted on the outside of the cell. But one of the really sneaky things that HIV does is it internalizes all the flagpoles so that the infected cell can no longer tell the immune system, hey, I have a virus, here's little bits of it, come learn what it is and come kill all the infected cells. HIV, it's incredibly complex and clever. I hate to admire HIV in any way, but really it's such a clever virus in all the ways that, all the tricks it has up its sleeve for how it can evade the immune system. But the cool thing about that, well, now that we're talking about NK cells, is that NK cells can actually detect cells that don't have those flagpoles, because NK cells know that it's probably a virus that did it to that cell. And so to me, that's one of the really promising things that you can try to use NK cells for as kind of a complement to killer T cells, for example.
Yeah, there's another advantage, and again, this was alluded to earlier when you were talking with Tim. NK cells work very quickly. In contrast to CD8 T cells, only a few antigen-specific, so CD8 cells that are targeting HIV proteins themselves, are made in the body. And they're made early, probably before one goes through puberty. If the virus comes in, those few cells have to react against those proteins and expand, proliferate, and just amplify thousands of times. They get tired when they're doing that. So in HIV disease, they tend to get exhausted. And so after a lot of chronic infection, with not just HIV, but other chronic viruses, the T cells are what we cleverly call exhausted. They don't work as well, okay? Natural killer cells are made all the time. Okay, now they can also get exhausted, but they're made fresh all the time. So it's a different army than, it's analogous to a T cell potentially being a military that's hired on day one, and 30 years later, it's the same soldiers. They're not going to fight as well. Whereas NK cells are coming in every day with fresh NK cells. Okay, so that helps. So there's multiple advantages. They can target different proteins. They could be fresher, different parameters for exhaustion. Yeah, so that's some of the advantages of using those cells.
All right. Now, one of the really complicated things that you're doing, because you, like I said before, you really have a whole bundle of complexity in this research grant that you're working on. But because we've talked a couple of times now already about these CAR NK cells, I figured let's start with that CAR technology. And I want to share the image of the, let's see, yes, this is the right one. So maybe you can talk us through this diagram, just so people get an idea of, you know, CAR, chimeric antigen receptor,
right.
What that even means.
Okay, so a CAR, which stands for chimeric antigen receptor, means you are engineering a receptor from two different bits. Okay, so it's chimeric because it's two different things combined together. And what that CAR is, which is shown kind of as that bronze-looking thing sticking out of the yellow cell, it has on the outside of the cell a receptor that will bind whatever the target is. Okay, and that can be engineered to be whatever target you want. The inside of that the CAR that goes inside the cell, which you can't see because it's inside the cell, has a signaling component that when the outside binds its target, the signaling component turns on the cell to kill that target. Okay, and if you mix and match different outside components, you can target different target proteins, target cells, okay, and kill different things. So what this picture is trying to show, and it's based on a tumor model, which is, it's very hot now in the cancer field with T cells. If you put a CAR into a T cell, it can target particular tumors, especially B-cell lymphomas. They get killed pretty easily by CARs. What we would like to do is put HIV-specific CARs. So those are molecules that can target HIV proteins on the surface of the cell, those little spikes that you were talking about, engineer those into NK cells to better direct the NK cell to kill the HIV-infected target. The rationale would be, if you put that in while the patient is just coming off of ART in a strategic treatment interruption or in a potential for kick and kill with a latency reversing agent, those cells that were latent are now going to be producing viral proteins on the surface quickly and that engineered NK cells should be able to attack it. Okay, so what we've shown in our models so far is the normal natural killer cell without engineering can affect the latent reservoir, can drop the rebound. And so now the next step is to test engineered NK cells to see if they're better. We're going one step further by also trying to engineer that into stem cells, which can then be turned into NK cells in the body. So you'd have a constant armamentarium of amplified, if you will, more effective NK cells.
So you've invoked cancer a couple of times, and we know that there are CAR T cells. There are even CAR T cells approved by the FDA. So they've been around since, well, they were approved in 2017. So, you know, that almost seems like it's old technology now. So CAR T cells have been used in cancer. What would make a CAR NK cell better than a CAR T cell?
Yeah, so one of them is the different timing of exhaustion, right? And especially if you go back to the approach that we're thinking, which is to put the CAR into stem cells, right? Those stem cells will constantly be making fresh NK cells that express that CAR for the rest of the life of that patient, okay? So the situation of exhaustion would be minimized because the cells are fresh. They're coming right out, but they're armed, right? They're primed to attack the HIV-infected cell. So we think that this could be a major advantage over a T-cell approach.
The way I understand it, CAR NK cells are less inflammatory than CAR T cells as well. Is that right? Is that your understanding?
Then CAR T cells, yeah, they're not designed to make the kind of cytokines that Tim was talking about, an inflammatory syndrome where lots of cytokines are made because T cells are activated. NK cells do make cytokines, but not to the same extent.
And some of the, you know, to the extent that CAR T cells can have, you know, a period of danger for the recipient of the CAR T cells, I think that's due a lot to the cytokines, this inflammatory kind of spewing out of all these toxins flying around. The other thing I'm intrigued about with CAR NK cells is the potential one day down the line to make them so-called off the shelf.
Yeah.
So, you know, with CAR T cells, you have to make CAR T cells specific to each and every person. But with CAR NK cells, maybe you could make them to where they're just about universally acceptable and you don't have to look for these close relations like Tim had to look for.
Yeah, I think cells that were not from the same patient would be effective at killing. The problem is they're going to generate graft rejection reactions. So you'd have to engineer those cells so they wouldn't be rejected by someone else's immune system. But there are technologies that people are working on to do that. So it's quite possible that you could do that, and you wouldn't have to have the cells as specific for a particular viral protein because they're going to attack sick cells or cells that are expressing any viral protein, really.
Yeah.
Yeah.
So now we've covered the CAR technology, but your grant doesn't stop there.
Yeah.
You're testing this in mice, as you told us, and you're using a technology called barcoded virus.
Yeah.
So I'm going to share a picture of that, and while you walk through telling us about barcoded viruses and what's so nifty about those.
Okay. So the idea behind a barcoded virus, and if you can see, that square thing is supposed to be the barrel of a syringe. Rowena drew this, so if it doesn't look like a syringe, it's not my fault. Okay.
Hey, my artistic skills, man, right up there.
Your mouse looks good. Okay, you probably copied that from someone.
Yeah, that was someone else's mouth.
I'm sure. So the little squiggles inside the syringe barrel are supposed to indicate viruses. And each of those viruses, if you look closely, has a little color piece on it. That indicates that we could tell each of those apart. So there's a way to make a genetic barcode that allows you to construct a viral swarm, if you will, where each individual virus has a different barcode on it or different color. So you can identify that virus and any progeny of that particular virus. Whereas a normal HIV, if you stick it in cells, they all look the same, basically, right? Here we can identify progeny of different individual viruses that replicate, okay? So the rationale behind this is if we use that to infect our humanized mice, those viruses will replicate. If we then put the animal on antiretrovirals, The cells that are actively producing virus will die. That's what happens. But those that are latently infected will survive and become a reservoir. That will be a very, very small number of cells, right? The idea behind that is each of those reservoir cells likely will have a unique barcode virus in it, okay? So now if we take the mice off antiretrovirals and we get rebound, the diversity of that barcode that comes out will tell us how many cells gave rise to that rebound, okay? How many infected cells. So what we're now doing is trying interventions like NK cells to see if not only do we lay the timing of rebound, but do we affect the number of barcodes that come out, which tells us the number of cells that gave rise to that infected cells that gave rise to that rebound are lower, okay? And actually we just had a lab meeting yesterday morning, virtually of course, where the data were shown where we pulled a bunch of our experiments together, adding NK cells, and it was absolutely beautiful. When we take the animals off antiretrovirals, the number of barcodes in the swarm goes way down compared to control animals.
Oh, good news.
So it's similar to what Tim was saying. He's trying to find tools in people. How do you measure an impact on the reservoir? You can't put barcoded virus in people. That even Tim wouldn't do that. You just can't do that. But you can do it in our model, okay? And so we can use that to study approaches that then eventually can be taken, not the virus, but the approach, taken to clinical trial to do things like Tim's doing.
Nice.
So we're preclinical.
Yeah.
And I think what's important for the people on the call to know is you need both arms. You need preclinical. Well, first you need bench science as well. You need preclinical modeling, and then you need in vivo testing in people to actually affect the cure. I think that
know what's the promising thing that is worth advancing to the clinic.
Yeah, and so we're the step ahead of what like people like Tim would try in clinical trials. So we try to find things, maybe they wouldn't work, maybe they would be toxic, and you don't want to do that in a patient. So you do that in our model, sort out what's the best strategy, and then
yeah. I mean, at amfAR, for sure, we're interested in clinical trials, but we also know the importance of keeping that pipeline going so that we'll always have the new intervention coming along that is the next thing that needs to be tried out in people. But on that note, I want to turn to a couple of questions that we've had coming in. So let's see if I know how to do this. This is a question for Tim. The infused NK cells found in lymph nodes, are they still cytotoxic?
I don't know. We're still doing those. Actually, that's the next step on our on our list of things to do. So in our trial, we took out a lymph node prior to the infusion of NK cells, and then after it was over. And we're just now setting up the cytotoxic or the functional assays to find out if those things are functional or can kill.
And we have another question for Dr. Schacker. How would transplanting NK cells from non-relatives complicate the process?
It's likely they would not persist as long and they wouldn't be as functional. So it's really, at least in the way we've done this study, it's really important to use the haploidentical or the related NK cells. But Jerry had made it a reference to a different approach, the off-the-shelf NK cells. There are a couple of companies now that are making these cells that you could use multiple times, and that might overcome the issue of persistence. And so, that's kind of the next step in what we're thinking about.
And so here it is. So this, Jerry, is clearly a question for you and from somebody who knows what they're talking about and what they want to ask. Which humanized mouse model do you use, and how's the baseline NK, macrophage, and CD4 development in this model?
That's a great question. So we use what's called a BLT mouse, which is fully reconstituted with human stem cells and thymus, and you get a very, very beautiful reconstitution of the immune system with two notable exceptions. Not a lot of NK cells, almost none, and a low number of macrophage and dendritic cells. So lower APCs. And that's actually, in our case, a strength, because now we can add new NK cells in and really build up the pool. We don't have a lot of naturally occurring NK cells in that model. So this gives us a a chance to see what the newly introduced NK cells do.
Tim, we have another one for you. How does the use of NK cells as cancer treatment differ from how it's used to treat HIV? You were talking a lot about the similarities, I think, but what would you see might be some of the differences?
Well, the presumably in cancer in people with refractory cancer that have not responded well to therapy, the tumor burden is probably much higher. And so that's one big difference. But that's probably it. I mean, the concept is still the same. You've got a cell that is not, you've got a cell that you can identify as wanting to eliminate. It's either not you, it's a tumor cell, or it's expressing HIV proteins. And so, you want to eliminate that. And the other difference is tumor cells tend to be quite active. And so they're expressing these proteins and they're easy targets. HIV infected cells, the latently infected cells, you've really got to kick those things to get them to present a target for the NK cell to work. So, and it's likely, frankly, that IL-15 alone may not be enough of a stimulus. We're going to have to, combine this with other things to not only induce the cell to make more target, but to help the NK cell clear the infected cell.
You said something, I'm going to say it anyway. It sounds peculiar, but I'm going to say it anyway. But you're reminding me that as hard as everybody knows it is to deal with cancer, there are ways in which cancer is actually easier than HIV. Along the lines of what you were just saying, in terms of targeting it, I mean, try to, just ask everyone in the audience, try to wrap your mind around that. We all know that cancer is a terrible thing and that a lot of people are working very hard on finding the best treatments and cures for all kinds of cancers. But consider that HIV is actually even more complicated than that. It blows my mind. Jerry, we have another one for you. Has barcoding to track individual viruses been used before?
There is a relatively recent a strain of SIV, which is a virus that infects non-human primates, that has been barcoded and is being currently used quite well. The non-human primates are a great model for studying similar things to HIV disease. There's some advantages of our model over non-human primates, in that one, we don't use primates. Two, it's much faster. So our animals are much quicker than what one can do in monkey models, which take a couple of years to perform. So there's some really nice studies with an SIV barcode being done. But to my knowledge, this is the first replication competent. By that, I mean replicating virus that can grow in an animal model that's HIV and barcoded, to my knowledge.
All right. We're coming up towards the end, but we have some other comments here that are not questions, but I'm enjoying reading them, so I want to share. I want to share some of them. Alejandro Mata says "me gusta," and we say "muchas gracias." Krista Martel says "awesome work. We love to hear about women and HIV cure research." And we have a comment that I'm not going to try to read because this is not a language I do. But we have some love from Brazil. So thank you very much, Brazil, for joining us. And, we are getting towards that time where we need to wrap up our conversation here, unfortunately. I've really enjoyed hearing from both of you. I think this work is fascinating. I know that we could talk for a really long time about that. Before I do that, though, I think it's intriguing, and I want to ask you both a question. You both have options, but you've both decided to turn to amfAR for support of these projects. So, Tim, I'll ask you first. What makes amfAR a good place to get funding from?
In projects like this that are a little bit more complicated. Well, not more complicated, but the amfAR has the ability to pivot quickly, to fund innovation in a much more agile way, I think, than some of the other major funding mechanisms. And so when you've got ideas like this where you're going to hey, I want to take NK cells from my brother and put them into this person. It was a little bit easier to navigate those waters. So it's the ability to be agile and move quickly that really sets amfAR apart in the funding world.
And Jerry, what made you turn to us?
Well, there's several reasons. Tim touched on some of them, but it's the ability of your program to fund innovation that would not be easily funded elsewhere. So projects that are more than just seed projects, but not really full-blown things ready to go, but quite important and innovative. And the other is there are aspects of our work, at least, that aren't easily funded by federal sources. And the fact that you're not a federal source does not have the same restrictions. And so we can do studies that we couldn't do on federal grants. So amfAR has been really great for us on more than one occasion for those reasons.
Great. Listen, the hour is upon us. I want to thank both of you so much for taking the time to join us. It really was an incredible conversation. We kind of just scratched the surface here. I want to thank everybody who's online submitting comments, very much appreciated, and some great questions, which I think, made our conversation that much livelier and more interactive. And of course, I want to really thank everybody on this call for your support of us, our board members and our donors, because if it weren't for your commitment, the work that we at amfAR do and the work that our grantees do just wouldn't be possible. So all of us are really just so grateful for that. Now, some of you, might have been to Meet the Scientist events before, and you know that we normally do these in person. So it's a little bit of a shame that we can't, we'll see each other and stand around chatting and, with each other in person, which I think is a lot of fun. On the other hand, if we're going to look for a silver lining here, this platform has allowed us to spread these messages and this content more widely. It's going to live on some of these platforms, so you can rewatch and watch your favorite moments again if you like. If you are able to, please do consider making a contribution to amfAR in support of our World AIDS Day campaign, in support of our grantees like the ones you've heard from tonight. We are going to put a lot of that towards supporting this program and some of the other programs we have going on at amfAR. If you're on YouTube, you can click the donate button there. You can see a URL on your screen that you can go to support this event, amfar.org/MTS. Please do. We are really so grateful for every way that you can support us. Thank you again to Jerry and Tim, and for everybody who has joined us here tonight for your time. Please have a great holiday season, and we hope to hear from all of you very soon. Thank you.
Thank you. Stay safe.
Thank you.